Hi everyone, I'm one of the 37 trillion cells that make up you, and today I want to talk about meiosis.
Most of the cells in your body are somatic or body cells and have diploid or the normal number of chromosomes. However, the cells that we care about today are your germ cells, which have half the number of chromosomes, and so they're called haploid cells. You may ask, why do we need cells with half the number of chromosomes? Well, the answer is that our germ cells, also known as our sex cells, are needed for us to reproduce sexually so we can create genetic diversity. Everyone has a different combination of DNA from his or her parents, so they are unique. Just look around you and you can see genetic diversity everywhere. In populations, genetic diversity is the reason species are able to evolve and survive. Germ cells have half the number of chromosomes as a regular cell so that each parent can contribute DNA and the offspring will end up with a normal amount of DNA. If this didn't happen, the amount of DNA people would have would double each generation. Every germ cell is unique and has a different combination of the parent's DNA, while each somatic cell is genetically identical.
Let's now learn how meiosis creates 4 haploid cells from a single diploid cell. The process of meiosis is split into 2 different processes called meiosis 1 and meiosis 2. Meiosis 1 is the splitting up of homologous pairs, and meiosis 2 is the separation of sister chromatids. At the end, there are 4 cells with half the number of single chromatid chromosomes. Let's slow down and take a better look at this. So let's say an organism has a total of 8 2-chromatid chromosomes in a normal somatic cell, and it is going through meiosis. During meiosis 1, the chromosomes line up in homologous pairs. Homologous pairs are 2 chromosomes that have the same set of genetic information. Then, the cell splits in half, creating 2 new cells. Each new cell has 1 chromosome from each homologous pair, so a total of 4 chromosomes. Then, during meiosis 2, each 2-chromatid chromosome splits. Each 2 cells created during meiosis 1 split again, so there are a total of 4 cells with half the original amount of genetic information. So now that you've learned a little bit about meiosis, let's learn exactly how it creates genetic diversity. The 3 processes that meiosis creates genetic diversity through are crossing over, independent assortment, and segregation. Crossing over is when chromatids from homologous pairs cross and trade genetic information. So, looking at this pair, which has the same type of genetic information but different variations, we can notice they have traded some information so that each chromatid is a combination of genetic information from both the mother and the father. Independent assortment is also involved with pairs of homologous chromosomes. It is when the pairs are randomly split into 2 different cells during meiosis 1. In a homologous pair, 1 chromosome came from the individual's mom and the other came from the individual's dad. In result, each germ cell has a random combinations of chromosomes from each parent. Finally, segregation is just the separation of the pairs of homologous chromosomes. It means that each pair gets split up so each cell ends up with half the pair. Woo! That was a lot to learn about, so let's review. You'll learn that meiosis creates germ cells and helps create genetic diversity through 3 different processes. Ultimately, meiosis is what helps make you an individual. Thanks for learning about meiosis with me today.